Hello everybody, I'm delighted to be here today. I'm going to take a slightly different tack than I do usually in my talks and focus this talk around the families of individuals with ReNU'd syndrome and how we can make progress and impact working together with them. So you've already been introduced today to Charlie in Rich Scott's talk in the plenary this morning. So Charlie is a 12-year-old. He has global developmental delay and intellectual disability. He needs help with everyday tasks, including eating, dressing, and toileting. He's completely non-verbal, and he needs routine and is very easily frustrated. Now, like many individuals with a similar diagnosis, Charlie went on a very long diagnostic odyssey. He had a battery of different genetic tests, but he was given no answers. And this is a quote from his dad, James, writing in the Sunday Times, saying, "Without a diagnosis, we had no idea of his prognosis, and so for the next 11 years, we were living in the dark, raising a child with no handbook, feeling alone, and blaming ourselves." That was until last year. So Charlie was a participant in the Genomics England 100,000 Genomes project. And last year, we, along with another team, gave a diagnosis to Charlie and his family. And we identified that variants in a non-coding RNA called ReNU'd cause a very frequent neurodevelopmental syndrome. Now, I'm not going to talk about the discovery in depth, because actually a couple of talks last year covered the discovery itself, but just to give you the highlights. So we found that variants in a very small region right in the center of this gene, so the gene is only 145 base pairs long. We found variants in a tiny 18 base pair region in the center of it, firstly in Genomics England, and this region is depleted of variants in the UK Biobank, and then collaborating with researchers worldwide in France, Germany, Australia, and the US, we identified in total a cohort of 115 initial patients. But what makes this finding particularly remarkable? Well, we estimate that there are around 100,000 individuals worldwide who are living with ReNU'd syndrome. Now, that's quite a lot for a rare disease, but just to kind of really highlight why that is pretty special here, I'm going to compare this in this table to the 3 genes that contribute the most diagnoses to the Deciphering Developmental Disorders, or DDD cohort, 10 years after it began. And these are these 3 genes here, ANCOD11, KMT2A, and ARID1B. And as you can see, the proportions contributed by these genes are not much higher than 0.4% of all NDD caused by variants in ReNU'd. But where the difference comes is when you look at the makeup of those different genes. So those 3 genes are all very large, over 9,000 base pair long protein coding genes. That's a lot of hit space for a de novo variant to occur to cause a neurodevelopmental disorder. So we can compare that to 145 base pair long non-coding RNA. And not just that, but 13 base pairs in a contiguous 18 base pair region of that non-coding RNA. This still completely and utterly boggles my mind. So what does this mean? Well, firstly, it means a diagnosis for Charlie. And Charlie's, so as Rich said earlier, if you haven't read this article yet, please go and dig it out. It is absolutely beautiful. And Charlie's dad, James, is a writer for the Sunday Times. And he wrote an article communicating what this diagnosis meant to him and his family. And again, these families tell us 2 key things. So firstly, that they blame themselves. And it's nice to know when they're giving a diagnosis that it wasn't something they did during pregnancy. It wasn't their fault. But also, they then get this community, and they can talk to people who are on the same journey as them. The bond fused over 1 million shared parenting moments, and they feel no longer alone. But rather than me telling you about the impact on these families, I hope I can get some of the families to speak for themselves.
This is Devin. We're in New York, and we have been searching for answers for 13 years. Getting the diagnosis of ReNU'd has enabled us to form together with other parents to start an organization that aims to advance research and renew hope for others all over the world.
Hi. We're Diana's family. We've been waiting 7 years to know about the ReNU'd syndrome. We're living in Poland. Knowing the diagnosis, it's given us the opportunity to connect with other parents and gain from their experience.
G'day, I'm Simon, this is Sarah, and this is Abigail. We're from Adelaide, Australia, down under. Abby got a diagnosis when she was 16, and getting a diagnosis has been really important to us because it removed a lot of questions we had before the diagnosis, and also means we're now part of a global community, doesn't it, Eddie? High five.
Yeah! Woo! Bonjour, je vous présente Thibaut. Il a 13 ans et demi. Nous habitons en France, au Relecq-Kerhuon, dans le Finistère, au bord de la mer. Cela fait 12 ans que nous étions en attente et nous avons eu le verdict fin janvier 2025. Nous sommes très heureux de faire partie de cette grande famille.
Hi, I'm Lindsay. This is Lars. We're in Virginia in the United States. We received our ReNU'd diagnosis in March 2024, and it's been life-changing for our family. It's really sort of changed our identity from being undiagnosed to being part of this community, but we're just so grateful to no longer feel alone and to have this renewed hope.
Hi, I'm Elliot and this is my little brother Samuel.
We're from the northeast of England and we didn't get Samuel's diagnosis till just for his 3rd birthday.
For us, having a diagnosis just meant that we had answers and a newfound community that we could belong to.
Hello, this is Cameron, and he is 16 years old.
We live in Florida, and it took us over 15 years to get a diagnosis for Cameron of ReNU'd syndrome.
Having the diagnosis of ReNU'd syndrome means that we are no longer alone.
Can you say hi, Cameron?
Hi.
So hopefully you can tell from that video that this has had a global impact. We're getting diagnoses around the world. There is now widespread testing for ReNU'd syndrome in many different countries. Here is just showing you the UK test registry, where this is now standard of care for individuals with neurodevelopmental disorders. Here is a map made by the wonderful families, where every dot on this map is a diagnosis of ReNU'd syndrome. This map simultaneously fills me with joy. There are so many dots on this map, but also with complete horror. There are so many gaps. So we as a community have a lot to do to try and get these diagnoses to all patients with ReNU'd syndrome. And given the need for genome sequencing or a targeted testing approach, there is a bigger healthcare disparity in ReNU'd syndrome than there are for a lot of different genetic conditions. So we need to work to try and fix this. But this is one of the core aims of ReNU'd Syndrome United, which is a patient advocacy organization that's now been set up. This is a not-for-profit in the US. There is also moves to try and set up a charity here in the UK. ReNU'd Syndrome United aim to fundraise, to increase awareness, to build that supportive community, and to mobilize research into their condition. And to fit a lot of these aims, they have their first family conference, which is a family researcher meeting, which will be in July next month. Hopefully I can get into the US because I'll be devastated if I don't make it. So as I've said a few times, one of the big impacts on these families is that they now have a community. Jessica, who was the first mom in that video, is an absolute super mom. She's incredible. She was one of the first families to be diagnosed out of the UDN in the US, and she set up this Facebook group. And I love watching the numbers tick up. For any of you that can't see it, there's now 446 family members in this Facebook group. We also worked alongside the families and also Sarah Wynn and Unique to create this patient information guide, which is now used by clinicians around the world to give information and support to families. What was also mentioned in Richard's talk this morning was the naming of ReNU'd syndrome. I learned a hell of a lot about the difficulties of naming syndromes over the past year. There aren't really that many good options. You might say, well, why can't we just call it ReNU'd associated neurodevelopmental disorder? Well, what then happens if we have a dominant and recessive form, or a gain of function and a loss of function form that are actually distinct conditions? This just doesn't work. And this is why we often end up with these eponymous names. So OMIM had created this horrendous acronym called NEDHAFA, which is basically an acronym of the symptoms of this disorder. And the parents, they hated it. They said it was stigmatizing and negative, and they didn't want their children to be defined by a list of symptoms. It also just doesn't always fit. So while a large proportion of individuals with ReNU'd syndrome are nonverbal, some of them actually can speak. So it doesn't actually speak to the disorder in a lot of individuals. So instead, we looked to this quote from Nicole Cedar, who's pictured here with her daughter, Mia Joy. And she said, as part of our initial press release in The Guardian, we like to refer within their family to ReNU'd as ReNU, a really clever play on the ReNU in the gene name, as their family is being renewed by this new information and hope for the future. So I then sent an email to OMIM. It was a very long email, and it had a hell of a lot of feedback from all the different families about how much they hated Ned Haffer and how much they loved ReNU'd syndrome. And then we managed to get this changed, and it now seems like this is stuck. The families have also been absolutely incredible at trying to raise awareness of ReNU'd syndrome. Why is that important? Well, to try and fill the holes in that map. So sometimes families can actually advocate for themselves and get testing if they think their child might have a particular condition. So in addition to James's wonderful article in the Sunday Times, there was a coverage on the national news in Australia, and actually only a couple of weeks ago, ReNU'd made the front page of the Washington Post, which I found out on the day and then scrabbled about trying to figure out how on earth I might find a Washington Post in the UK. I didn't succeed. I got a PDF. But these families now have an insane amount of hope. But that's hope that I think we have to temper. But I think there is really good reason for that hope. So the discovery of ReNU'd syndrome comes at a time where we have the hope that we could develop a genome-targeted therapy. We heard just a few weeks ago the story of KJ, who had a personalized CRISPR therapy developed in a very short time frame and delivered in Philadelphia. There is also the well-known story of Mila and Tim Yu and Mila's mum, Julia, who has spoken at this meeting before. So we now have the tools that can help us to try and develop a therapy for these individuals. ReNU'd syndrome is also pretty special that it actually hits a frequency threshold that it's actually of interest to pharmaceutical companies. And there are at least 2 companies that I know that are interested in trying to develop therapies for ReNU'd. However, the reason for the tempered hope are, I kind of guess, threefold. So firstly, we have a hypothesis of what the mechanism of the disease is, but we still don't know that. So we've got a lot still to do to understand deeply what that mechanism is in order to try and treat it. And also, can we tolerate even increase or decrease in levels of ReNU42, which might form the basis of a treatment? Is this even treatable? It's a severe neurodevelopmental condition. How much of that disorder has progressed too far at an early stage for us to actually develop an effective treatment? We need to do detailed natural history studies to understand the history of the disease and how it progresses, to understand if a therapy could be effective. We also need to know how do we tell if a therapy actually works? Can we find a biomarker? There's some real promise here because this is a core splicing factor. It actually disrupts global splicing. Can we use that splicing signature as a biomarker to inform development of a treatment? In order to do this, we're actively working on developing cell line models, both from patient cells, but also using CRISPR and iPSC lines. And also we now have a mouse model of ReNU'd developed in collaboration with Jack's Labs in the US. But I just want to put a little plea here that if we want to succeed in this aim of developing a treatment, we all have to work together. And that is the researchers, the pharma companies, and the patients and their families. It's also been absolutely incredible to see how research into ReNU'd has progressed in just a year. Here is just a quick screen grab of research papers. This is probably not comprehensive, but this is what a year's worth of research looks like. It's pretty incredible. I just want to mention one of those very briefly, partly because it's the paper that we've done as some follow-up work, to just add a little bit of science in my comfort zone into this talk. So in this paper, we worked with Greg Findlay, who's a group leader at the Crick Institutes here in London, to perform saturation genome editing of ReNU42. So what does this mean? This means that we made a series of different edits. We introduced variants across the entire gene into a haploid cell line called HAP1 and tested the impact of each of those variants on cell survivability, hypothesizing that if a variant was deleterious to cell survival, it would be more deleterious in a patient. And this is basically what this data looks like. So each dot on this plot is a different variant, and along the x-axis we have the 145 base pairs that is the entire gene. Now, as you can see, the majority of these variants are above that black dotted line. This means that they don't have a large impact on cell survivability. We call them neutral. Whereas we get some variants that drop below that dotted line that we say are significantly depleted and likely to be deleterious. The vast majority of those variants you can see are in this central section shaded in gray, and this is the region that we know is critical to ReNU'd syndrome. And all the variants that are in red, which are variants that we've actually seen in individuals with ReNU'd syndrome, fall below this black dotted line, as they're significant in this assay. So this assay performs very well to help us identify variants that cause ReNU'd syndrome from those that are likely benign within this region. What that means is that if we now have a new patient walking to the clinic that has a variant that we've never seen before, we can give a relatively confident estimate of whether that's likely to be disease-causing or likely to be benign. We can also split the variants in this region into two different categories based on whether they are only moderately depleting in this assay, so moderately deleterious, shown here in yellow, or if they're more severe, they have more of an impact on cell survivability. And what this allows us to do is actually these scores seem to separate out patients on an axis of severity. So to tell you that in a bit more of an understandable way, if we look at individuals with these less severe variants, over 95% of them can speak sentences or have fluent speech. And that's in comparison to over 90% with more severe variants being completely nonverbal or speaking only single words. So it's actually quite amazing how much this assay, how well this works. But what some of you may already notice on this plot, I'm glad it didn't skip ahead this time so it didn't spoil my punchline, is there are actually some other variants that on this plot that look to be important. And actually, we were initially confused by these because they deplete in this assay, but they're in blue on this plot, which means they're observed in the general population. And Greg very quickly said, well, perhaps this is because these are, we're using a haploid cell line, so we only have one copy of the genome. So perhaps this is causing a recessive, perhaps these variants are recessive. So actually what we then found out was that when we looked at these variants, we found that there is a recessive NDD that's also associated with variants in ReNU'd. So variants in completely different regions to the ones that cause ReNU'd cause a recessive syndrome, which is much, much rarer than ReNU'd syndrome. It's the gene that just keeps giving. So we'll add that to the growing list of snRNA-associated disorders. So ReNU'd was not the first snRNA to be associated with disease. The first was ReNU4 attack, which is a U4 equivalent in the minor spliceosome, which splices a subset of introns across the genome, about 0.5%. And variants in this and another minor spliceosomal snRNA, ReNU12, cause recessive phenotypes. But since we discovered ReNU'4 there's also been two additional major spliceomal snRNAs, ReNU22 and ReNU5B1, which have spoken about at different talks this afternoon that have been associated with neurodevelopmental disorders. But ReNU'4 at the moment is the first to have been shown to be involved in both recessive and dominant NDDs. But what this is really important for as well is, and I think Rich alluded to this a bit in his talk this morning, was that not only does this add hope to patients with ReNU'd, but the fact that we're still looking at these data and we can still make these discoveries that have such a large impact so many years after the patients have enrolled into the study actually gives them a lot of hope that they might be next, that there might still be a hope for them to find a diagnosis and join a community like the ReNU'd family. So I couldn't end this talk without flashing up a load of pictures of these wonderful patients, and also another advertisement for the family conference in New York next month, just in case anybody wants to come along. And I want to finally finish with a massive thank you. So I get to stand up here and talk about all of this, but there's actually so many people that have been involved in this discovery. We couldn't make discoveries like this without the participants who so kindly, and their families, who so kindly donate their DNA to research. This discovery was made because we transitioned from looking at a single patient to looking at many patients in tandem, and we can identify the variants that are in common. There are also so many staff, researchers, and within Genomics England who support and make all of this happen. So thank you very much to all of you. You're all incredible, and thank you very much for listening.